Welcome to one of the most misunderstood phenomenons in biology, which is the idea of diffusion. In class, I usually do an experiment where I will take a beaker or show a video of a beaker like this, somebody putting a drop of ink or something into it and watching the ink kind of, spread out. And then I ask the question, well, how did that happen? And students tend to use big words like, move from a high concentration to low, or osmosis, or things like that. But when I ask the fundamental question of how did the blue stuff know, how did it get the energy to spread apart? How did it know where to go? A lot of times I hear crickets. So this is actually the idea of diffusion. And diffusion, as you probably know, is a movement of some sort of molecule from a higher concentration of itself, see where it's way more concentrated, to a lower one. And when I always ask the question, well, how does it do that? Most people really can't answer the question. And the answer is very, very simple. Basically, what happens is, here's a barrier, by the way. So this is just like, a glass of water on this side and maybe a glass of Kool-Aid on that side. But if there's some sort of membrane they're allowed to go through, what happens is these molecules bump into each other. And so if you see back here, that's really what's happening. These things are bumping into each other, and now that they have more space to spread out, that's the power that they use. This is a passive transport system, diffusion. It doesn't require any energy. The molecules are literally bumping off of each other, bouncing around, and therefore spreading out. And that's how they do it. So you can kind of see here that this is what would happen if you were to have some sort of membrane between this and they were both allowed to pass, that eventually they'd keep bouncing around, that the concentrations would be equal on both sides. It doesn't mean that once half of the red's over here and half of the white's over there that they stop. It just means that the mathematical likelihood that if one moves over here, another will move back, they tend to be the same. So here's an example you've probably seen in A&P or other classes that kind of shows you that idea. And this is a red blood cell. And of course in here, this is a really important word to know. A solute. And basically a solute is something that gets dissolved. Water tends to be something called a solvent, which does the dissolving. So if any of you guys are familiar with, for example, what's that movie called? Wizard of Oz. Thank you. Sorry, I wasted 4 seconds. At the very end of it, Dorothy threw water on the Wicked Witch of the West. So in that particular analogy, the water would be the solvent and the Wicked Witch of the West would be the solute. She was the one that was being dissolved. So based on this, solute is what I call anything that's really not water. It's stuff, it's materials, it's sugars, it's salts, it's things like that. So given the idea of how this happens, if this cell has a membrane that's semi-permeable to some stuff, you'll see here that the cell maintains its normal size and shape because for every relative amount of water that leaves, the same relative amount comes back, and that's called isotonic. Now if we look at something called a hypotonic solution, of course hypo meaning less and tonic meaning solution, we'll see that there's less stuff on the outside and there's more stuff on the inside. And so there's kind of a little bit of a lie that I'm going to tell you right now that's actually true 99% of the time, but not absolutely true, is that water tends to go to where there's more stuff. And stuff is anything that's not water, as far as you're concerned. So if we take a look here, we have a little bit of solute on the outside, and clearly this cell has way more salts and sugars and solutes on the inside. Water tends to go to where there's more stuff, and so therefore this cell is going to swell because of that. And the same is true in the other case. If we add a concentration around the cell where there's way more stuff, water tends to go to where there's more stuff. And in this case, there is more stuff on the outside of the cell. The cell is going to lose water, hence a hypertonic solution. And so that's generally kind of how that works. And understand what's going on here, that this is actually osmosis. When I talk about the diffusion of water, that's osmosis. So if I were to basically, be in the room and spray some cologne in the side of the room, slowly all of you in the room would smell it. That's diffusion. But if the stuff that it is that's actually doing the diffusion is water, then we call it osmosis. So osmosis is just diffusion where the substance that's diffusing is water. So let's try a couple of these to kind of get the idea back. So in beaker A, you have basically a semi-permeable bag. Boy, isn't that a weird looking bag. And it's floating in or sinking in this container of salt water. And there's 10% salt water in the beaker, and there's also 10% salt water in the semi-permeable bag. What's going to happen? The answer is nothing. The bag is not going to gain or lose weight because the same amount of water that's moving in is going to be moving out. So this is a little different situation. So here you have a bag with 10% salt solution in it, and it's in a beaker that's filled to the top with 2% salt. So basically you have to ask yourself, where is there more stuff? And the answer is there's more stuff in the bag. Water goes to where there's more stuff, and so therefore that bag is going to gain water. There's going to be a net influx of water. And you may be asking yourself, why? Why is there more water on the inside? And the answer is very interesting, is that water being polar, meaning that part of it is negatively charged and part of it's positively charged, it basically is attracted to the salt. And of course, in this case, understand that chemically speaking, some salts can leave the bag XYZ. But even if we made this sugar, let's just keep it simple. If there was sugar in these containers, that would be a better example. But the water is going to be attracted in and out, but it's going to stay inside because it's attracted to the parts of the sugar or the salts. And of course, it will depend on the size of the salts as to whether or not it actually can go across the membrane. But in this case, we're making an assumption that nothing in the bag can leave the bag. Only water can go in or out. And the idea is if water starts diffusing in, it's going to stay there because it's going to be attracted to the salt. This is the opposite situation. So what if we have a semi-permeable bag that is not permeable to this type of salt? The salt can't leave the bag. We also have 30% salt solution that the bag is sitting on the outside. So the question here is, if water is the only thing that can move across this, which way is it going to go and why? And if you pick that the actual bag is going to lose mass, lose water, you are right, because water goes to where there's more stuff. And in this scenario, you can see that the environment has more stuff. There's more stuff outside of it than on the inside. So although water is going back and forth, once some of the water leaves, it's staying on the outside because it's interacting to some degree with that solute, be it salt, sugar, something like that. So hopefully that helped to give you a little better example. The last thing we're doing here, last 20 seconds, we consider this isotonic. We consider this environment or the solution surrounding it to be hypotonic, and we consider this environment or this solution around it to be considered hypertonic.